Was earlier. So the plan for today is to give the lecture that I intended to give on Wednesday. So we're going to finish up the topic of behavioral ecology and then go ahead and start the next topic, which is population ecology. Lecture 23, which I was hoping to give today, I'll record as soon as my voice improves a bit more, probably either later today or tomorrow sometime. I'll upload that as a podcast and you can listen to that. That'll be the rest of population ecology. Just to bring this to your attention, I'll offer an extra office hour last week to make up for the one that I had to cancel yesterday. So next week, in addition to having the regular office hour on Thursday at 2 o'clock, I'll also have an extra office hour on Tuesday at 1 o'clock. Both of those office hours, per usual, will be in room 5248 McGaw Hall. I hope to see some of you then. I was asked to make this announcement in class, and that is let you know about a course that is being offered next quarter. This is a one-unit class, pass, no pass, and I thought it might be of interest to some of you if you're interested in going into the healthcare, one of the healthcare professions. So this is an opportunity to have discussions with healthcare professionals about their experiences in compassionate healthcare. If you're interested in this flyer and all of this information is on the Clubs and Other Opportunities tab of the course website. You can check that out there. When we left off last time, I promised that I would give just one example of a kind of behavior, and so the behavior that I've chosen to share with you is the example of associative learning. And this is a type of conditioning in which an organism is taught to associate two stimuli with each other, and these are arbitrary stimuli, meaning that they don't necessarily go together. One of the primary scientists who's associated with advances in associative learning is Ivan Pavlov, who won the Nobel Prize in Physiology and Medicine for his work in digestion. So he is particularly well known for his classic experiments in training dogs to associate stimuli such as the ringing of a bell with another stimulus, which was being fed. So I've got a little cartoon to show you here. Let's see, I think it's starting. Yes. You know, so this shows the sort of cartoon form, how the training actually takes place. The training for the organism is that the animal, in this particular case a dog, is exposed to the stimulus such as the ringing of a bell, and then the dog is fed. And after these two stimuli have been after the dog has been exposed to these two stimuli in association, after a while the dog does learn to associate these. Now, how can you tell if the dog in fact has learned to associate these two stimuli? And the answer is that when the arbitrary stimulus is again given to the animal, in this case the dog will show some sort of response that allows one to infer that in fact it is expecting the second stimulus, namely salivation in the absence of the actual steak appearing for the dog. So that's an example of associative learning then. Classic experiments. But as behaviorists do such experiments, they're interested in the underlying causes of the behavior at two fundamental different levels, and these two different levels are described as proximate and ultimate causation. They have a specific meaning. Proximate causation refers to short-term causation, meaning how is the behavior is occurring? What is the underlying physiological or genetic or neurological hormonal basis for the behavior? What is so this is really quite different from what is meant by ultimate causation, which is the term used for an explanation of behavior looking for a longer-term underlying reason in the context of natural selection, meaning what would be adaptive about this particular behavior. So this is a really quite different kind of way of asking questions. One is short-term and one is long-term. So let's have a little practice here and see if you can figure out which is which. We'll do this as a clicker question. Any answer counts. So consider the case where you have a female cat in heat. If you don't know what in heat means this female cat is sexually receptive. Okay? A female cat in heat urinates more often and in many places. Male cats congregate near the urine deposits and fight with each other. Which of the following would be an ultimate cause? Ultimate cause meaning, in the definition that I just went through, ultimate versus proximate causation. So which of the following would be an ultimate cause of the male cat's response to the female's urinating behavior? That is, why would this be adaptive? Why would this occur in the context of natural selection? I've given you 4 different possibilities here. A, the males have learned to recognize the specific odor of the urine of a female in heat. B, when the males smelled the odor, various neurons in their brains were stimulated. C, responding to the odor means locating reproductively receptive females. Or D, male cat hormones are triggered by the odor released by the female. So those are 4 different possibilities for you to ponder. If you're not quite sure, good time to chat with the person sitting next to you. Be friendly. If you don't know them, you can say, hello, my name is, what do you think? I'll give you 20, 30 seconds or so to ponder this, and then I'll shut this down. Okay, everybody should have clicked in. We'll see what you guys think. Well, almost 2/3 of you got it. Nice job. Okay. Now, some of you might have been looking for clue words like natural selection or adaptive or something. That, of course, could be a dead giveaway or it could be a red herring. What's maybe a bit more reliable is to think about the actual context, meaning of the sentence in front of you. So, for example, if we look at A, the males have learned to recognize the specific odor of the urine of a female in heat, that is describing a learning behavior. It's not saying why this would be an adaptive response. This one is clearly a physiological response describing the neural basis for the response. This one, as 2/3 of you correctly identified as a correct answer, is a correct answer because it identifies an adaptive response. That the reason why this might be expected to be selected on in natural selection is because it is advantageous to be able to locate sexually receptive females if you're a male. And then D is also an example of a physiological response, so that would be considered proximate causation. So great job. If you didn't get this one correctly, then that would be a good thing for you to review the difference between proximate and ultimate causation. So within the broad category of behavioral ecology, there are several different categories or types of lines of inquiry, types of questions that are asked by behavioral ecologists, and we're just going to consider a couple of those. The first one being the area of foraging. This is usually referred to as optimal foraging, and this phrase, optimal foraging, is used to describe the consideration of benefits and costs associated with a specific behavior of foraging. So what is foraging? Foraging means looking for food. Out moving around, looking for food and acquiring food. It may surprise you that there are costs and benefits associated with certain kinds of foraging behavior. It might help to consider some of the costs and benefits to being out there foraging. It's much easier to think of the benefits. After all, if you're out looking for food, then the most obvious benefit is, of course, actually finding food. But what costs are associated? What harm is there in wandering around looking for a food item or a prey item or the right kind of plant that you happen to eat? Well, as you are wandering around, you are in fact burning up calories. And so in a situation where food is actually limiting, this can be quite a large cost. So this is an unnecessary wasting of energy looking for food, if in fact it's unnecessary. Another possible cost associated with foraging or moving around is exposure to predators. And so as you're moving around looking for food, then this is when you in fact could become food for somebody else. So let's now consider some of the experiments that are discussed in your chapter. We'll start with this one here, which is diagrammed in figure 53-3 from your textbook, which is now in front of you. What this shows is a couple of petri dishes full of maggots, that is, immature fruit fly larvae. And so these little objects right here are supposed to represent the fruit fly larvae. And as noted, an undergraduate student who was working on a research project looking at the behavior of these maggots as they were crawling around in the petri dishes noticed that some larvae tended to move more than others. And she reared up these fruit flies and crossed them with each other after separating them into two groups, which she called rovers, meaning those that tended to move around, and sitters, meaning those that tended to move around less frequently. And she noted that when she crossed rovers with other rovers, that their offspring tended to move around more, and when she crossed sitters with other sitters, their offspring tended to be less likely to move around, and therefore this is an indication of a heritable component to this particular kind of behavior. Now, that's all very well, but how does this relate then to possible costs and benefits? As pointed out before, moving around unnecessarily is a waste of calories. So when would it be adaptive to actually be a rover then as opposed to a sitter? If we move this figure then off to the right, so we have room to write next to it on this slide, let's consider some sort of condition that would change, that would have an effect on fitness, because ultimately that's what we care about if we're considering the adaptive significance of these kinds of behaviors. So consider then a case where food is limited or population density is changing. So if we consider population density here on the x-axis, where we have high population density over on the right and lower on the left, this will be related to food availability, which I'll put in a different color here, so availability, there we are. Food availability will show the opposite relationship with population density, such that the more larvae you have packed into the same area with the same amount of food, the more food will be limited to any of those individual larvae. Now, would we expect any difference between the rovers and the sitters in terms of how fitness would relate to population density? Well, yes. One difference you might expect, for example, is that if the population density is very high and food is low, that rovers would have higher fitness under those circumstances because they are more likely to find new food sources. We would actually expect the opposite kind of relationship for sitters, in that under conditions over here on the left, where population densities are low and food availability is very high, that a sitter genotype, phenotype and genotype, would be likely to have more fitness because those individuals are not wasting calories wandering around looking for food. This is an unnecessary behavior. Whereas in conditions of high population density or low food availability, rovers, in fact, would be expected to have higher fitness than sitters because of their behavior, they'd be more likely to come across new food patches than the sitters who were moving around less frequently. And so these kinds of differences in fitness, depending on environmental conditions, is one of the reasons why such diversity in different kinds of phenotypes and genotypes is in fact seen in natural populations, where different phenotypes and genotypes are particularly adaptive under one set of circumstances, but the other one is in fact more adaptive under a different set of circumstances. So from this kind of behavior, or from this kind of relationship between fitness and being either a rover or sitter, then you should be able to figure out if after many generations, under constant conditions, would you expect the rovers or the sitters to be preferentially represented in the population, depending on what the circumstances were. I'll leave that as a thought problem for you. Let's go on to the other example, which is somewhat more natural than just considering fruit flies in a petri dish, and that is considering gerbils, which you may know from pet shops, but gerbils in real life actually do live in the real world, and so this is a study in one of the deserts of Israel with some gerbils that are native to the desert there. So this study looked at gerbils in their natural habitat in the desert, and the scientists investigating gerbil behavior were interested in how their foraging patterns would be modified by exposure to dangers such as their predator, which is the owl. So they set up an experimental plot as shown here diagrammatically in figure 53-4 from your textbook, which is now in front of you, where there was a big plot in the desert as shown here, and it was divided into two. The gerbils could freely cross from one side to another. Over here on the left-hand side, it's labeled as control subplot. That meant that the investigators didn't do anything to manipulate that side. It was just ordinary desert. And then on the right-hand side here, these scientists would have one kind of manipulation or another. And specifically, the kinds of manipulations that the scientists did was either to allow owls to fly over without actually eating the gerbils, but just simply flying over so the gerbils could actually see them. And there was also another treatment where there was owls flying over and there was extra food supplied to the gerbils. They eat seeds, and so it was extra seeds as shown here on the right-hand side. So let's look at the results then and how the scientists then interpreted these behaviors. Here are the results shown right here to remind you of what the color coding means on these bars. Here's the legend here. So orange stands for the control and the red stands for the treatment. Let's start over here on the far left-hand side where we're considering the case where on both sides of this large plot there were no owls flying over, nor were there extra seeds. Both of the halves of the plot were exactly the same, and so therefore it should not surprise you that what the results were. If you look at the y-axis here, the y-axis is gerbil foraging activity. There was no difference on the two sides of the plot, which is an important control, of course, to demonstrate that there's no sort of intrinsic differences between the two sides of the plot that were not obvious to the investigator. So, gerbil foraging activity is the same on both sides if there's no manipulation. What happens if owls are allowed to fly over on the right-hand side of the control treatment? Then the results are shown right here. Foraging activity goes up on the control side, so presumably some of the gerbils had left the dangerous side where owls were being spotted and moved over onto the other side, and there was less foraging going on the side where the owls were flying overhead. So this shows then a behavior that could reasonably be inferred as being adaptive to try to avoid the presence of predators. But what's particularly interesting here is the results shown on the far right. So what the scientists did now was, in addition to having owls flying over, they were interested in the extent to which they could offset the cost of an increased risk due to the the danger of owl predation, with offset it with a benefit. The benefit being an increased food supply. And they found that, in fact, by supplying an additional 4 or 5 grams or so of seed per gerbil per hectare, that was the amount they found that was necessary to offset the danger then. So if on the side that had the increased owl predation, if they also supplied extra food, then both sides of the manipulated plots were equally attractive to the gerbils. So this kind of clever manipulation or clever experiment then allows one to actually ask the organism to calculate and compare the benefits and costs, because it's a little bit difficult to do that a priori. It would be very difficult for the scientists, in the absence of such information, to try to predict the extent to which just simply giving gerbils extra food would in fact offset an increased danger. So these kinds of experiments are very useful for understanding the behavioral responses of these organisms in their natural environments. Okay. I'm going to go ahead and leave the general area of food now and getting food, foraging, and change to the second major category, which is sexual activity. So we're just going to go through one example from this section of your textbook, and that is an experiment which is diagrammed in figure 53-5 from your textbook, which is in front of you right now, and this involves the sexual activity of some male lizards. The genus is Anolis, as described here. So in these lizards, the males show a particular kind of display during their courtship and mating behavior, which is shown here in this figure at the top. So the males bob up and down, and they expose a structure underneath their throat, as shown here, which is brightly colored. So this is a visual display for the females and indicates to the females that this male is receptive and interested in mating with the female. Now, the graph down here in the lower left shows the sexual receptiveness or readiness for sexual reproduction of both males and females as a function of month during the year. So the months are on the x-axis here. These letters respond to the first letter of the month, right? So this would be November, December, January, February, and so on. So it's sort of starting here with the beginning of winter, and then spring, and then summer, and so on. So we've got the readiness for sexual activity being described as the size of the sexual organs. So this is very different from what one sees in humans, for example, which are not seasonal breeders, and so you wouldn't get this kind of graph for our particular species. So notice in this particular species, and the breeding season is during the late spring and early summer, and both males and females are graphed on here. So the red curve shows the relative size of testes, so this is showing the sexual activity or readiness of males then. The testes are the gonads of the male. And the blue graph shows the sexual readiness of the females, right? The relative size of the ovaries, the gonads and the females. So they are smaller during the non-breeding season, and then they become larger as breeding season approaches and then occurs. So the scientists were very interested in what is it? What are the proximal stimuli that are causing the changes in the gonads of both the males and the females? Now the data that is actually shown in this figure is just for the females, so we'll consider the females here, although both males and females undergo changes as shown here. Well, it was pretty clear that it was hormonally driven, but what is it that's ramping up the sex hormones, the testosterone in the males and the female sex hormones in the females? What is it that's actually causing changes in those hormonal levels? The scientists decided to evaluate the factors, that is, looking for proximate causation, by the experiment, the result of which is shown here in figure 53-8, which is in front of you right now. This is a somewhat complicated experimental design, which is why I'm taking some time to explain this in lecture. So this actual experiment was performed during the winter when the females are not typically sexually active. And in fact, as a control, down here in the purple line right here, this is the control where the scientists were evaluating the sexual readiness of the females that were just left outside in that natural habitat. And you can see that these females were not sexually ready at all. The y-axis here is as percentage of females with mature follicles. That's follicles in the ovaries. You can think about that as the percentage of females who are sexually receptive and able to sexually reproduce. So out in the natural environment during the same time period then, the females were not sexually mature at all. So that's one kind of control. So these were females just left outside. The rest, all of these females, and as I pointed out, both males and females change, but this particular set of data is just for the females. These are all the females that were brought inside into the laboratory, and as they were brought into the laboratory, the scientists made it spring, sort of a fake spring inside the laboratory. And how did they do that? They increased the temperature, and they also changed the light cycle. So they increased the length of daylight. So the time when it was dark was shortened artificially inside, and the time when it was light inside was lengthened artificially. So all of these different lines then had the same changing conditions. So notice that this is not the kind of experimental design that one would use if one was interested in teasing out the differences between light and temperature, because all of these females had both an increase in temperature and an increase in daylight length. In addition to changing the temperature and day length, these scientists changed the social context for these lizards. And so a female lizard, some female lizards were paired with a single male and put in a container. Other females were put with a group of males in a container. Other females were put in a group of males, but the males had been castrated. This means that their testes had been removed, so they were no longer producing testosterone, so they were no longer capable of sexual activity. And then these other two groups here were females that were either put with females, or the females were just left alone in their own container. And so you can see a couple of different things from this graph, right? One is that just simply being brought in and warmed up and having a change in daylight is already having an effect on bringing the females to an increased state of sexual receptiveness here. If you compare these lines, whether they're blue or red, with the purple line, you can see that at weeks 3 and 4, these females are closer to being sexually active than the females that were left outside. But in addition, notice that these groups are separating out from each other, and in particular, the females that were in the vicinity of males had a different response. They became more sexually active more rapidly than females that were not around males. And so the actual proximate cues have not yet been identified for this, but that's the logical interpretation for these kind of data. Okay, so that's the proximate cause, right? Exposure to temperature or presence of males or changing daylight temperatures. You should also consider, well, what is if we're considering the ultimate cause for these kind of changes in the female with changing sexual activity or receptivity, then what would be an ultimate cause? Why would it be adaptive for a female to change her sexual receptiveness depending on seasonality or presence of males. So I encourage you, as you read through that section of the book on sexual reproduction, that you consider proximate and ultimate causation for the variety of different kinds of experiments that are being considered. I won't spend any more time on that right now. Okay, we're going to change gears a little bit here now and make the switch to considering population ecology. So in considering population ecology then, we're interested in understanding the factors that affect the distribution and abundance of organisms. And so this is normally done, of course, in the context of the natural environments of the populations, and the way that the populations are usually quantified in their environment includes such factors as density. So this is basically, how many organisms per unit area. There's also interest in looking at the spatial pattern. If you go and you look at the organisms, how are they arranged in space? The age structure of the population, how many organisms are old, how many are young, what is the population structure in terms of age? And then the overall total population size, the total number, is also of interest. This is different from density, but is obviously related to it, right? So meaning that the total size of the population as opposed to what kind of area is that population packed. So let's start by considering patterns in space. So as population ecologists look at the distributions in space, they can sometimes gain some understanding about interaction between individuals, or something about the environment, or something about how the organisms got there in the first place. And the three major categories are shown here, along with a diagrammatic representation. So the way that spatial distributions are usually characterized are clumped. So this one right here would be considered clumped, where individuals, where each dot is supposed to represent a single individual. So in a clumped distribution, all individuals are very close to another neighbor, although you can have more than one clump. In a uniform or even distribution, the individuals are spaced out in a regular manner as shown here. And then in a random distribution, as shown here, the location of any given individual is completely independent of the location of any of the other individuals. So each individual's position is independent of the positions of the other individuals. Now, if somebody's not used to looking at these kinds of spatial patterns, then they often look at a random pattern and think of this as being clumped, because after all, if you look at a random pattern, you can certainly see that some of the organisms or some of the individuals are closer to each other than they are to other ones. It's certainly not evenly spaced. But if one randomly puts dots on an area like this, then simply by chance, one can end up with dots that are closer to each other than others. To actually differentiate between these patterns unambiguously, then one actually has to measure the distances, and then one can use mathematical distributions to actually identify it to one or the other. We will not be using the mathematical methods to differentiate between these. We'll just look at them as overall patterns like this. We won't go into the underlying math. Let's look at an example of a clumped spatial distribution. So here's a picture not from your book in front of you right now. So this shows, for an example, a cluster of sea stars. And in the intertidal, they're often clumped. And One can get clumped behavior because of a couple of different reasons. One is because the environment is patchy, and so the organisms are responding to the patchiness of the environment, such as the availability of prey items, by clumping together, and that's exactly what's happening with these voracious predators, these sea stars, which are clumping together to take advantage of some prey right here. You can also get clumped distribution patterns of individuals if, depending on the kinds of social interactions of individuals, where some organisms do tend to clump together. Social interactions don't always lead to clumped distribution patterns. So here's actually an example where interactions between individuals leads to a uniform or evenly spaced distribution, as shown here. Penguins and some other kinds of birds are territorial during certain periods during their life cycle, and during their aggressive encounters where they're setting up territories, then they tend to form a space around, individuals tend to form spaces around each other, and they space themselves out in the environment as shown here, such that there is a uniform spacing. So usually when one sees a distribution like this, it's because organisms are having some sort of aggressive interaction with each other. And in random distributions, it may surprise you, are actually more rare in nature, and an example, though, of a random spatial distribution would be the examples shown here, where the pattern of these weeds are just simply driven by the wind-blown seeds that blew into this field and then were randomly deposited, and where they landed, that's where the weeds grew up. So this could then lead to a random pattern, which is driven by the initial random placement of the seeds. Okay. So regardless of how organisms are distributed in the environment, those population ecologists often want to know, in addition, well, how many organisms are out there in this particular population? And it should not be a huge surprise to you to think that it's usually completely impractical to actually go in there and count every single last individual in a population. So typically, methods are used to make estimates. And there are a number of different methods that are used. We're only going to go through two, and we'll go through these in order. First, we'll consider that one could just simply take a sample of a population, a subsample, and then extrapolate to the population as a whole. And the second method is called the mark and recapture method. So let's start by considering how one could take a sample of an environment and then extrapolate. So I'm going to use this as a visual backdrop to discuss this. Imagine you're out in the wild and you have some way of taking the spatial extent of some organism, which here will be these herbivores here roaming around. And if one wanted to try to estimate the total number of individuals, but one wasn't able to actually count every last individual, then one could simply divide the space into squares and then go in and count a subset of these squares. If in fact the subset of squares that is being counted is representative, then one can use that to estimate the overall total. So this is usually fairly intuitive to students. So let's go ahead and practice this and see if it's intuitive to you. We'll do this as a clicker question then. So here's the example. Consider a habitat, and we're going to divide it into 100 squares. So 10 rows by 10 columns. We've got our grid superimposed on our habitat. We're interested in making a population estimate for some critters that are within these 100 squares, but we can't count all of them, but we can count the individuals in 5 squares. After we count the individuals in these 5 squares, we then know, because we just counted, what the average is, and here's the result. The average of each square is 2 individuals, okay? So the total habitat is 100 squares. We counted up the individuals in 5 squares. We got a result that the average number of individuals in each square was 2. So from this, you should be able to figure out how large the population is, right? So if you're not quite sure, this would be a good time to chat with your neighbor. I'll give you 20 seconds or so to figure this out, and then click in, and then I'll see what you guys think. Okay, I'm guessing all of you have figured it out by now and clicked in. Again, I would expect this to be fairly intuitive for most of you. So, all right, let's see. Oh, fantastic. All right. All right. Most of you got it. Excellent. Okay, those 20% of you or so who didn't. Okay. So how would you figure this out? Well, we've got 2 individuals per square, right? At least according to our estimate, the 5 squares that we were able to count. And then we have a total of what? 100 squares, right? So we just multiply then 2 individuals per square times our 100 squares. 2 times 100 is 200 then individuals. This is our guess for the overall population size. And it should make intuitive sense to you that this estimate will only be as good as our 5 squares are representative of the total. And so in fact, if we happen to pick 5 squares that were under or overrepresented, then our estimate would either be an over or an under representation of the true population size, depending on which way it went, right? Okay, if you got that one wrong, then this would be a good thing to practice in office hours or a discussion group. Okay, let's go on to the next method. So this method is called the mark recapture method because it describes very nicely and clearly what one does in order to use this method. So this is a method that can be used for organisms that can move around. You can't do this for something like plants, right? So how does this work? Step number 1, you have some population. It's very difficult for you to estimate the total number, but you are able to at least capture some individuals. So maybe you're able to capture some ants outside the ant nest, or you're maybe able to capture some birds in a bird netting trap or something. Anyway, you capture some individuals and you mark them in some way that doesn't harm them, and then you release them back into the population. M1 is just simply an algebraic notation for indicating how many individuals you actually captured and marked. So for example, it could be 10 if you caught 10 ants and marked those 10 ants and then put them back near the nest. So step number 2 is you return these marked individuals to the population, and then you wait for an appropriate amount of time. The appropriate amount of time is dependent on the biology of the critters that you are dealing with. But you wait until it's a good guess that the individuals that you return to the population are now thoroughly mixed in the population. And now you go back and you take another sample of individuals, and this only works if some of those individuals in your sample are in fact marked. So these N2 and M2 then are algebraic notation for describing how large your sample is and how many of the individuals in that sample are actually marked. From this, you can calculate the population size. This seems like magic until you think about what the underlying logic is for this. Okay, let's consider what's going on in the population, because I think that's a lot easier to wrap your mind around. Consider or assume that capital N indicates the total population size. You don't actually know what that number is. That's what you're trying to figure out. However, you do know that within that population, which has a total of capital N individuals, that M1 are marked because you marked them. So in the population as a whole, the proportion of marked individuals is M1 divided by big N, right? This is the proportion of marked individuals in the total population, of the population as a whole, right? Does that make sense? Right? Big N is just the total population, and M1 is how many you marked, and therefore it's got to be true that the overall proportion of marked individuals in the population as a whole is M1 divided by capital N. Right? Now, if you waited long enough, and the marked individuals, in fact, did mix themselves thoroughly into the population as a whole, then it should also make intuitive sense to you that as you take another sample, if that sample is representative of the population as a whole, then it should have exactly the same representation, exactly the same proportion of individuals that are marked. So, for example, if you were able to mark 10% of the population, when you initially captured and marked those first individuals, if now 10% of the population is marked, if you collect 10 individuals, then one should be marked, right? Because 10% of the overall, of any sample should be marked. So what is the actual proportion in this sample? Well, we had a total of N2 individuals, right? That's how many we had total in our sample, and we know how many were marked because that's M2, right? So the sample that we took after returning the marked individuals to the population weighting, then the proportion in our sample is M2 divided by N2. If in fact the proportions are the same, which they should be, hypothetically, if the population was thoroughly mixed, the marked individuals are randomly dispersed in the population, then these two fractions should be equal to each other, right? So we have just simply small m2 over n2 equals m1 over big N. We've got 4 items in this equation, but only one is unknown, and that's big N. That's the one we want to solve for. So if you cross multiply, then you can get m off by itself here, and you have m equals small m1 n2 divided by m2, as shown here. So you know all 3 of those items on the right-hand side. You can plug them in and estimate the total population. This is pretty extraordinary and powerful thing that one can do. So let's try it out. Okay, you knew that was coming. Let's have some practice in doing this one then. Okay. So you've got your population, you're interested in estimating the total population size. You grab 100 individuals, 100 ants running around outside the nest, and you mark them all with some non-toxic paint that doesn't harm them, and then you return them to the population. Then you wait long enough that they seem to be thoroughly mixed back into the population, and now you take another sample of 100 individuals, and you notice that 10 of those 100 individuals are marked. So from this, you should be able to figure out how large is the population. So I'll give you 30 seconds or so to 45 seconds to figure this out, chat with your neighbor, and then I'll check back in with you. Sheesh, you guys doing it in your head?
What?
What's that?
Oh, I already clicked it.
Oh, you did? Oh, you guys figured it out pretty quickly. Too easy. Okay, clearly too easy. You guys have already figured it out. Good. Click in. Let's see what you guys think here. Has everybody clicked in? Okay. Fantastic. Excellent. So more than 80% of you got that right? So for those of you who didn't, how can you do this? Figure out what's going on in the sample, what's going on in the population as a whole, right? 30 more seconds, guys. In the sample, we've got 10 out of 100. Population, 100 out of n. Set those equal to each other, right? Solve for n. Great. Have a great weekend, everybody, and we'll continue population ecology on Monday.